mention of identify your target, and uh, that's what I first said yesterday. You have to first identify your target. Take for example now in your previous study, probably you discovered a mutation or you saw a mutation that has been reported in the study. Like I used example yesterday, I said we're going to be using the HBB as an example. So if there is any mutation in the HBB gene and you want to convert back to its original state, you have to first identify the target site, which is the first thing here. And you can do that by probably getting your sequence from the database or sequencing your sample. The sequencing of the sample will give you the order of the nucleotide. So depending on which one you want to do, it's fine. And the next thing for you to do is to design your guide RNA, which we did yesterday. And I said, well, today we're just going not to repeat much of the things that we've done. I'm just going to show us how you can now pick your best guide RNA. The purpose of this class is not to suddenly groom you to start designing. No, that is not the idea. The idea is to give you the background because just like you are in your BSc now, you are doing because I want to believe it is not only gene editing you're doing in this program. So you have other fields you're doing bioinformatics, you're doing molecular biology as a whole, you're doing other aspects, you're doing writing. So it is after this program that you can now decide which one do I want to venture in, which one do I want to specialize in, which one catches my interest. Um, so that's why I said this program is not to quote and unquote train you to just suddenly become bigger. No, it's just to give you that background that you can leverage on. So if you don't get as much as you can get, don't feel bad. Just identify where you think at the end of the program your interest lies, then build it up from there. We don't just arrive suddenly. So, and that's why I don't want to make things much more complex for you guys. I just want to give as easy, as straightforward as explanation I can give, so that when you now want to decide, okay, you want to be, what I say, venture into genome editing, then we can now take it up from there, then climb into the advanced or what I say the intermediate ones. So we designed the guide RNA yesterday. Today we're going to now see what criteria gives us the best guide RNA to pick. So initially I thought I was going to teach us how to design your construct, but trust me, we don't want to do that now because, like I said, this is not a master class. It is not a master class, so we can't do that for now. So just get just get the concept, and then we're good to go. So after designing your guide RNA, of course, I told us we're going to clone it into a plasmid. We're going to clone it into it depending on how you design your experiment. Sometimes you can clone into your Cas9 plasmid because the Cas9 is an enzyme and it has been incorporated into a plasmid. So you can clone your guide RNA into the plasmid. And if you're going to do the cloning, the type of cloning you're going to be using, the type of cloning you're going to be using is the infusion cloning. So when you do that, then you cannot transfect. You can, of course, you're going to use bacteria for the cloning, maybe E. coli, depending on the strain you want to use, DH5 alpha, top 10, HB101, depending on the strain. So far, it's a good cloning bacteria, a competent cells. So you can clone you can clone your plasmid inside so that you can have a multiple copy of them and increase the efficiency. And then you extract the plasmid DNA, then you can now transfect it into the cell line or mice or parasites or whatever model you're using. So using electroporator. So you're using electroporator to put them you're using electroporator to deliver the plasmid into the organism so that they can get in. If you're using mice or you're using mosquito, especially for people in doing gene drive, use micro injector. So electroporator is not just the only way. If you're using cell line, maybe a cancer cell line, whatever cell line, or maybe even a parasitic cell line, you're going to use electroporator. But if you're going to be targeting like Drosophila, melanogaster, if you're going to be targeting zebrafish, if you're going to be targeting that has to do with embryo, you're going to be using microinjector so that you can deliver the plasmid through microinjection. So, irrespective of whatever you choose to use, all depends on the model you're going to be using. So there is no standard. Just know the model you want to use, and then select the transfection method you want to go with. So after you've transfect your plasmid inside your organism, the next thing you should do is to test. What are you testing? You're trying to check if your transfection works. So then you can, depending on the downstream analysis you want to do, but the first thing you have to do first, and also depending on the model you're using. If you're using a cell line and transfecting inside the cell line, sometimes it gives them like a week for them to recover, because at that moment so this is how it works, right? You're trying to stress the organism. So imagine you want to give me something now and you made me to fast. I will be weak. So if you give me something if you stress me at that point, I might want to die. So you will now give me time to recover so that I can express whatever you've given to me. Okay, so the same idea works. So at the point of, because when we say electroporation, the electro means electric field. You're transfecting them with an electric, so they're going to be electrocuted, quote and unquote. They are going to be stressed electrically. So you give them time to come back to that normal state. You give them time to come back to their normal sense. Even when you want to do a transfection, maybe during your cloning process, the bacteria you're going to be using, either you can as well use electroporation, or you use heat shock method, where you can use your water bath or heat block to heat shock the bacteria so that the plasmid can go into the bacteria. At that same time, you ought to give them a food, which is called the SOC or the LB media. The idea is just that you want to stress them. So immediately you stress them, you have to be giving them food so that they can recover. Because either using it shock, either using electroporation, you are giving them a stress. So you have to give them time to recover back. The same thing goes to your transfection. If you're using transfection, if you're using electroporation, you want to use electric to introduce the plasmid to them. So when you do that, you have to give them food. In this case, now maybe cell line, you have to be giving them a media in order to sustain them. So it now depends. They are coming back now depends on the model. For cell line, 2 3 days they might recover, but for parasitic cell line, it might even take 3 weeks for them to come back to normal. So that's why I said the transformation efficiency is sometimes crazily low. So it might imagine you give it another 3 weeks, and you now found out that none of the cell line took up the plasmid. So that means you have to repeat your experiment again. So this is how I said the CRISPR-Cas9 experiment. Your art is not ready to be, if you don't build immunity against outbreaks, you can't do CRISPR-Cas9 experiments. So your art just have to be rigid. So, I'm just trying to explain all of these things to us, not to just give us theory alone, but to give us the practical sense of what is going on in the lab. So when you transfect, you give time for the organism to come back to life, or would I say to recover, then you can now do your test. You know, the first thing might probably be your phenotypic, uh, would I say, accessibility, or you just want to confirm phenotypic validation. You want to confirm the phenotypic means what you can see. You are seeing the cell line. You want to confirm if something has changed. Okay. So the next thing is to confirm at the molecular level, which you can do your PCR to confirm if the plasmid has been integrated into the cell line, or into the mice, or into the zebrafish, depending on whatever you want to use. So when you do your PCR to confirm, then to just be sure very well, you can now do sequencing, or you can first do your RFLP first, which is the restriction fragment length polymorphism, using the restriction enzyme to confirm. So there is, in your plasmid, you have different cloning sites that is being guided by a restriction enzyme. Let me see if I can get one now. In just a minute, let me show you what I mean by a plasmid. Because what can we see at the moment?
We are still seeing the schematic diagram.
Okay. Are we seeing another slide now?
Yes, please.
Okay. Okay, so this is a plasmid. Let me take this one away. So this is what how a plasmid looks like. So all this you can see different enzymes here, and these are the different regions in the plasmid. These are the different region in the plasmid. So this is my 5HR, this is my 3HR. So you can see different enzymes. So there are different cloning sites. So the point I'm just trying to bring out here is that when you want to do your confirmation, then you can now use any of these restriction enzyme to digest your amplicon that you got to be sure that there is a digestion, because the enzyme you're going to use, take for example now, this is let me look for one enzyme. This is SAC2 enzyme. So if I use SAC2 enzyme to digest the what I said, the PCR product, I should be able to have multiple band, because the enzyme should be able to recognize this site, because the plasmid is already inside the cell line. So if I use this particular enzyme on the PCR product, I should be able to see a digestion, so that the enzyme recognizes the site. Remember the function of restriction enzyme. It cleave a DNA at a recognition site. So the enzyme should be able to recognize this particular site, provided the plasmid is already in the cell line. Once this SAC2 recognizes the site, it's going to cut. So that is another level of confirmation in the lab. So the third level of confirmation is now you doing, let me go back to my previous slide. The third level of confirmation you want to do in the lab is to do sequencing. So you can now do sequencing. So can we see the previous slide now?
Yes.
Oh, thank you. So the third confirmation you want to do is it third or fourth? I don't know. The next confirmation, I should say, is your sequencing. You want to sequence to make sure that there is no mutation. You want to sequence to make sure that everything you put inside this model is well there. There is no addition, there is no subtraction, there is no artificial motivation that is going on. So once you confirm everything, you confirm your editing, by sequencing. So you can now, once your sequence, you for just to be double sure of what you're doing, and depending on the experiment, if you're doing a mutagenesis, once you sequence, it is the next thing for you to do is to blast. So you're going to blast. That's why I say you can't do CRISPR-Cas now without bioinformatics. You're going to blast against your original. You're going to have a wild type control. Of course, there's no experiment in the lab you want to do without having wild type control. Remember, this class is not just your typical theory class. So that's why I don't want really to be going into it. That's why I'm giving us a practical perspective. So I'm not just preaching what is in the textbook. I'm telling you based on my own experience, which is to give you the practical perspective of it. So when you're done, you want to do blasting. So you can now So you will have your sequenced then editing has been, you've confirmed the editing by PCR, you've confirmed the editing by RFLP, you've confirmed even the editing by sequencing. Aware type control. The white type control means you didn't do any gene editing experiment in that one. It's just your normal white type. It's a control for you. So you have to blast them against each other. But one of them will serve as a query. When our bioinformatician mentor will take us, I don't want to go deep into his own parts. So you're going to see one as a query, you're going to see one as the subject. So you're going to blast them against each other, blast N using nucleotide against nucleotide. So the blast is just to compare. Take for example, if what you do is an SNP or SNV, a single nucleotide variant, you just only changed a one nucleotide. So you will use your blast to confirm if the one nucleotide in the wild type is now different from the one nucleotide in the, what I said, the mutated line now. So if your control is GAC, your wild type that you've changed should be GTC. So it means the A has been changed to T. But if you're still saying GAC in your white in your mutated line, it means your experiment did not work because it shouldn't be the same as the white type. So that is another level of confirmation in the lab. So that being said, so once you are done with your screening part, then you can now go on to do your functional studies. All these ones I've mentioned now are just confirmation. So once you are confirmed that everything is good, then depending on your functional studies, maybe you want to do a drugs assay. You want to you want to confirm the effect of that mutation, especially in the cancer cell line. You want to confirm the effect of the mutation, so you can expose the wild type to a particular drug, expose the mutated line to a particular drug. You want to see if the mutation is conferring a drug resistance in the organism. So that's can help you to now do a comparative study. So that is a functional assay. You can as well do, if you're doing for cancer, the one I just been mentioning is for maybe parasite line. If you want to do for cancer, you can do colony formation, you can do spheroid growth, you can do invasion assays to just compare and contrast your control against your transgenic line. So all now depends on whichever functional assay you want to do. And if you're doing that, you're going to do a Western blot. Of course, you want to see that if the protein is actually expressed. And the Western blot is sometimes part of the confirmation as well. I forgot to mention of it. It's sometimes part of the confirmation. So that there is a particular protein called the 3HA, which is a tag, a glutenin tag. So it helps you to confirm if your gene editing as well works at the protein level. So you're going to see it because, of course, it's a protein that calls for a gene. So you're going to see it not just at the DNA sequencing level, you're going to see it at the protein level. That is what the Western blot is going to do for you. And then once everything is confirmed, then proceed to your functional assay, do your functional assay, then analyze your result depending on what you're doing, and you're good to go. So that's just would I say, the total overview of what is going on in the lab in the practical sense, not just the theory, CRISPR this, CRISPR that, no, 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 blah, blah. And one thing I will tell you is that CRISPR that you know in theory is totally different from the CRISPR you will meet in the lab. I told us yesterday, if you want to do this kind of experiment, get yourself to being bullied by the experiment. So the CRISPRs, if you see some people talking about, sometimes I just laugh, not out of pride, but out of what I see, I was just, these people are really talking. I don't want to say ignorant. And it's not ignorant per se, they're just trying to be enthusiastic about it. Oh, CRISPR-Cas9, especially if you've not really done anything on CRISPR-Cas9 and you're kind of preaching it, I'm not saying you don't preach, but the point I'm just trying to bring out is that what the person that is only limited into the theoretical part, you will notice the difference to the person that has actually done something on it, because it is when you get to the lab you will know that whatever you have been saying, I've been there before too. I used to talk about CRISPR theoretically. It was when I got to the lab and noticed that, oh, this thing is actually different, because I've done different CRISPR experiments, I've done on what maybe when they get to the application, I will talk more about that. So that being said, theory part is different, totally different from practical sense. So that is just what is going on in the lab space about the CRISPR-Cas9 experiment. Before I move on, I want to be sure that we are on the same page. I want to believe we can hear me. Okay. So, now, let me just quickly finalize what we were doing yesterday so that we can I can go to the core part of today's class. So this is what we have yesterday. Like I said, my initial plan was to take us through construct, and I realized that it's going to be a whole lot of. It's not something we can just do 2 3 days online training and say we are no. It's better I don't go there rather than piling up confusion for most of us. So that's why I said if you graduate from this mentorship program, now decide what you want to do. Everybody cannot do CRISPR. Everybody cannot do gene editing. If you now decide what you want to do, then we cannot be taking off. So I just have to limit whatever I'm going to teach here to the bare is it barest minimum they call it now, so that we don't end up confusion for ourselves. So like I was telling us yesterday, these are the criteria you will use to pick your guide RNA, your target sequence, your genomic location, which I said is the most important. If your if your sequence is if your gene of interest, or would I say the target size is around 32 base pairs, then you can see these one, you can see 36, you can see 49, you can see 50, you can see 66, depending on the genomic location. And I made mention of the strand yesterday, which I said it can be on your sense strand, and as well it can be your antisense strand. The plus is the sense strand, the minus is the antisense strand. And I made mention of GC content yesterday, the same way you have a GC content in primer, in primer design. So you're gonna talk about the GC content as well here, which should be ideally 40 to 60%, which is down the range. And we talked about the self complementarity, which should be zero. The self complementarity means the ability of the DNA to of the guide RNA rather to form an hairpin, to form a loop to what I say bind to itself, which is not good because it's going to lead to off targets. And then we'll talk about the MMO, MM1, MM2, MM3. So the MMO ideally should be one. The MMO means at least your guide RNA should recognize a particular site. It shouldn't be zero and it shouldn't be more than one. It should be that particular site you design your guide RNA for is what it should recognize. So it should be one. Your MMO should be one. Your MM1 and MM2 ideally should be zero in as much as is possible. They at least should be zero. Like I said yesterday, sometimes you don't get to see what looks like a perfect. So you just have to work and optimize what you have. That I'm just trying to give us for ideal standard now. It should be one. And then your MM3 is of no, would I say, no relevance, so to say. Is not as important, it's not as critical, if I should put it in that sense. It is not as critical as the MMO, MM1, MM2. The most critical one is your MMO. And I talked about the efficiency, which I said is going to help you to now see how efficient the guide RNA would direct the Cas9 to cut. So let's just hypothetically pick one example now. Um, Let me see. Let me just look for a guideline that looks okay so that I will just tell us how you can do it. So we don't bore us with too much complexity, all those nonsense. They're all nonsense. So, let me just pick the one that looks reasonably okay. Mm, okay, so we can see coming.
Please, am I the only one still seeing the schematic diagram rather? Sorry.
Are you for real? Oh God, so sorry.
Yes.
So sorry. About now?
Yes, thank you.
Yeah, of course you can. All of the things I said are on the screen, so don't worry if you didn't get to see them. You can see the target site, you can see the genomic location, you can see the strands, you can see the GC content, you can see the cell complementarity, you can see the MMO, MM1, MM2, MM3 efficiency. Those are the things I said, so they are not abstract. So even you were not seeing it when I was seeing them, so at least you can see them now. So I'm just saying I'm going to just hypothetically look for a one to just do an example for us. Like I said, this platform will not permit us to do something as complicated as it is. So it's just for a background knowledge. So it's not to turn us to a gene editor over the night. So let's try and look for this. I'm just trying to look for someone. Okay, so let's just see this guy. So let's see this guy. So this guy is in position 170. Like I said, this is just an hypothetical design. So I just want to show us how you can now take it off from here. So I see this guy now. Let's say this is the guide arena I want to pick. The location is 170. It is on the same strand. The GC content, although it is 40, it is 35. Like I said, there is sometimes you can't get what you're looking for. I mean, I've used the guide RNA, which is would I say the 30% GC content, and they still work. So all depends on how you can optimize your experiments, how one can cover for another one. So you might not all the time get the best. So 35 is still relatively acceptable. Self complementarity, which is very important, is zero. Mismatch is one, which I'm good to go. Mismatch zero, meaning the guideline should be only binds to one site, which is very fine. MMO zero, fine. MM2 zero, fine. MM3 15, I'm not really bothered about that. And efficiency is 60.19, which is averagely good for me. So I will copy. You can either click on the sequence on the guide RNA to just give you more information, gives you where it binds in the inside your sequence. So just click on it and it comes to this interface. So okay, let me just copy the guide RNA again now. So this is my guide RNA. Then I will go to my sequence that I've copied. Then press find, then paste the guide RNA so that I can locate where it is. Sorry. Okay, so this is where it is now. So let's assume the GTG I want to target is here. I think I made mention of GTG yesterday. So let's assume this is my hypothetical target site now. New target site. Then that 20 nucleotide is going to be in between your C-terminal end of your uh, pre-guide stuffer. When you see the plasmid is always labeled pre-guide stuffer, that is where your guide wants to come and infuse on. On the upstream of the pre-guide stuffer, you will copy 20 nucleotide. On the downstream of the pre-guide stuffer, you copy 20 nucleotides. So that means your C end, your C terminal end, and your N terminal end, 20 nucleotides, so that your guide RNA can be in between them. So that is what you will now send for synthesis. So they can the same way they synthesize your primer. You see the same way you're going to get your guide RNA. Then probably it's going to come in a lyophilized form. Then you make it a solution. Then proceed to whatever you want to use. So that is just how to design your guide RNA. Now you can go ahead and just practice on your own with a little understanding you've gotten. Know the gene you want to target. Just practice with different sites so that at least you can use that one to know how to pick. Don't place emphasis on the target site for now. Just emphasis should be on how do you get the best guide RNA so you can randomly pick different sites. Random, like I said yesterday now, or like today now, this is the site I want to target. I want to change this GTG to GAG. So the guide RNA I'm picking is, and another thing you need to you need to be conscious of is that your guide RNA should be closer to your target site. That is very, very much important. At least maybe 3 to 10, if possible, 3 to 8 base pair away from your target site. If it is that close, it makes your work easier. If it is that far, it might probably lead to would I say an off target. So it should be able to be making it to, like, these are now my experiments as a possibility of being successful because the guideline I found for it is closer to my target site. So my efficiency is still good. So meaning my work has the possibility of being successful because the target side, the palm side is what is going to now activate the Cas9 to cut what is there. So that is all about the guide RNA design. Get your gene, practice. You can go to NCBI, just download random gene, get the sequence, follow what I did yesterday, copy the sequence into your Word, then change your font to Korean New. Like I said, this is just my own way of aligning them. Then identify your 5 UTR, identify your 3 UTR, your start codon, and your stop codon. Then just hypothetically randomly pick a target site, then know how to perfect your designing of the guide RNA. That should be the emphasis for now. I would have loved to take us through how you now design your construct that includes your donor templates and all of that, but trust me, it's going to be a complex journey. I wouldn't want to do that for now. When you graduate, like I said, from the mentorship program, when you now want to venture into genome editing, then it can be taken up from there. But for now, this is just a general class. The same way they will teach us general biology, general microbiology, general biochemistry in our school. When you now want to do your masters, you can specialize. That is what we just do here. So that will be all for design of guide RNA. And so now let's move on to today's class, which I said is going to be going to be talking about application of the gene editing. We're going to be talking about the troubleshooting in the lab. What do you do when you have some field experiment? We're going to be talking about the future perspective and ethical concern. So that is what we'll spend the next minutes to be talking about now, which is our today's focus. So, the gene editing, like I said, it's a tool. It's a genomic tool that is being used in different fields, both in medicine, both in agriculture, both in different contexts. As a matter of fact, I've been privileged to use both of. I've been privileged to use the same CRISPR-Cas9 in agriculture, and I'm privileged to be using it currently in medicine. So I'm not just saying what I read. I'm telling you what I've done. So it's not just about the theoretical part, like I said. So in the medicine application now, please can we see slide so that it won't be as though I'm still showing Chrome.
Yes, sir.
Oh, that's fine. So in the medicine part, it has been used as a gene therapy, which it corrects a defective gene causing an inherited disorder such as the sickle cell muscular dystrophy. And I think the gene I used to do an example of the targeting of the DNA of the guide RNA design is the sickle cell gene (HBB). So the gene therapy has been used as the sickle. What would I say? The CRISPR-Cas9 has been used. And a typical example of it, we did a workshop recently in the University of Lagos about gene editing hands-on workshop. And one of the guest speaker is Jimmy. I wouldn't know if many of us know him. Is a Jimmy something like that. He's an advocate for CRISPR-Cas9 technology because he is a sickle cell. He shared his struggles, he shared his pains. I mean, I think he even clocked 40, was it yesterday or something? So he shared the struggle he has faced for the past years of his life. Of course, you know how it can be to be a sickle cell patient. So, but as of 2019 thereabouts, he was one of the first people to be treated with a CRISPR-Cas9 therapy. They call it Cas-GV. So the Cas-GV helped them to correct this particular disorder. So it turned him from a sickle cell person to a normal person. So you can imagine that. And the guy is living fine. Even after 5 years, he clocked 40. I think he shared on his LinkedIn yesterday. He clocked 40 yesterday. 5 years now is still very living fine. No more pains, no more sickle cell, related sickness and all of that. The guy is fine now. So you can imagine this is a real life. We're not talking about theory now. This is a real person that has been transformed with the CRISPR-Cas9 technology. And we have a cancer research as well. Many, as a matter of fact, my supervisor, my master supervisor, is current MSc student, and I think one of his PhD students. They are currently working on in mice, and they're trying to target a multi-drug resistance gene in mice rather. They are going to induce the mice with the cancer. Of course, do some cancer induction to the mice so that they can be cancerous, so that some of the genes can be cancerous, and use the CRISPR-Cas9 to target them. They're going to treat them with a drug, so use the CRISPR-Cas9 to target the multi-drug resistance gene in the mice. I think the approach they're going to be using in their own case, I think they want to use the non-homologous engineering approach. Like I said, all depends on your objective. So I think that's what they are trying to do. So I'm trying to give us while I'm backing this up with a real life example. I don't just want to sound theoretical. Oh, it can be used in cancer research, it can be used in gene therapy. No. So that's why I'm trying to bring a real life experience to each of the example I'm giving us. So you don't just see it as though, they are just he's just saying it based on what is online. I'm giving us a real life experience with what is going on. And then the third one is infectious diseases. Of course, this is what I'm currently doing right now. I'm working on the malaria parasite. I'm trying to study the parasite survival strategy to anti-malaria drug resistance. So, and I'm using the CRISPR-Cas9 to do a functional assay. If I'm doing a conditional knockdown, in this case now, I'm trying to repress the particular gene to study its effect. I'm going to have an endogenous gene and I'm going to have an ectopic gene. Endogenous gene means the gene that is naturally present, and the ectopic means what I say the artificial copy of a gene, which might probably contains the mutations I want to study. So I'm just gonna do a comparative study, a functional study between the ectopic and the endogenous. So I'm going to use the CRISPR-Cas9 to introduce the plasmid into the parasite cell line. So that is what I'm currently doing at the moment. Like I said, I'm backing up each example with a real life experience, so that I don't just bore you with theoretical. And then for drug discovery, the genome edited cell line and animal model for rapid testing of new drug. When I was in the US, the lab I was trained, they are using their own for drug discovery. They are into malaria drug discovery, and they're trying to use the CRISPR-Cas9 to identify a novel anti-malaria target. In order for them to, because it's going to give them a new idea for, let's do a conditional knockdown, because those genes, you are doing conditional knockdown because the gene are essential. You can only do mutagenesis, side-directed mutagenesis, if the genes are not essential. You just want to knock out the gene and bring anything. But if the gene is essential to the organism, that in the case if there is any mutation, they might die, you don't need to do precise mutagenesis. You just need to do a conditional knockdown. And that is what they are doing over there, because the gene is essential to the parasites. So they're going to do a conditional knockdown to repress the gene, study its action. Can this gene be used to develop a new drug? It's going to give them a clue about new anti-malaria drug discovery. So that is what that is being done. And then personalized medicine, which is the tailoring treatment to patient genetic profile. This is the only one that is still looking as if it is not feasible yet. However, different research has been done to just make sure, okay, can we use this CRISPR to form a personalized medicine? We call it a precision medicine, so that each person can have a treatment that is in respect to their genetic profile, which I believe in years to come, we're going to go to that level. So, what about in agriculture? Because it's not just in medicine alone, it's been used in agriculture. Like I told us the other time, I've been fortunate, or would I say privileged, to use both in agriculture and both in medicine. And one of the things is crop improvement, enhancing drought tolerance, pest pathogen resistant yield, and shelf life, which is where the idea of GMO comes in. You know, I know there are so many debates around GMO now, and I'm not going to be talking about so that we don't spark any debate for now. So it's all fine. Everybody will always have different perspective for things. You know, I'm just trying to tell us that the CRISPR-Cas9 has well been used in the agricultural sense. And during my MSc project, I used the CRISPR-Cas9 to optimize a microbial enzyme production. I worked on lipase. My colleague worked on one of my colleagues worked on amylase. The other colleague, I think the last two colleagues worked on protease, and then another one on cellulase. And we have varying results. In my own case now, I noticed that when I did the knockout of that particular gene, a laxi gene, it gives rise to because I did comparative study after I've edited the particular gene inside the bacteria. I use both of them to ferment potato peel, and I noticed the edited one produces more lipase enzyme compared to the unedited, or would I say the white type E. coli. That is the one that the CRISPR-Cas9 is not used for them. And for other, I think one of my colleagues had the same result too, but the other one of us had, one of us, I think the person that worked on amylase, she observed not really different results between the edited E. coli and the unedited E. coli. Seems to be just, relatively the same. While the guy in the cellulase, its own is opposite of what we got. So the unedited even produce more higher than the edited. So it's just a comparative study, so in order to balance the microbial enzyme production, and that's the real life experience as well. So there's about fortification, enriching food with vitamin and mineral. We have livestock breeding. We have climate change benefits, and we have food security as well, which is to ensure sustainability of production in order to meet the global demand, especially meeting the SDG rather, SDG goal. Please, before I continue saying my nonsense, are we still together? All right. So,
Yes, sir.
thank you for that feedback. I just want to be sure. Okay, so the next thing I'm going to be talking about is application in industry and an environment, which already made mention of my experience. It can be used for microbial engineering, your bacteria for, because that is what would I say do, would I say did rather. That is what I did in the lab. It's not just theoretical. It's something I've myself done, and one of our paper has been published. The other one is in preprint stage, and hopefully it get published soon. And this work has been presented both in Nigeria and in Denmark. I think I presented mine in in Nigerian. What's the name of this organization? College of Nigeria. I presented my work there last year, and this same work, this lipase on, which is what I did. My supervisor earlier this year, he was the one that this CRISPR medicine conference, and I wrote the abstract, which was accepted because that time I tried and presented is one I go less for my training. So the work has been presented in the both, what I said, the local and the global stage. That's to tell you the importance of technology in different fields, not just in medicine. It's all round. It's employed all round. And then we have synthetic biology on converts the waste into energy, and that's still related to what I did. Of course, I use potato peel waste. So I use this as a substrate, so that my engineered, or what I say modified E. coli can act on it and produce an enzyme, or whatever you want to produce. And then the next one is environmental solutions. I wouldn't know if I mentioned of gene drive earlier. So the gene drive, unlike your normal gene editing, per se, but in this case now, you're trying to control an inheritance pattern. In gene editing, you're trying to probably introduce a mutation to solve a problem, maybe to to use as a therapeutic approach, you know. But in gene drive, it's not basically about therapeutic now. It's about the community, it's about the environment. And a typical example of the gene drive is malaria mosquitoes. So I'm just going to break it down so that it's easier for us. So one of my mentor in public health wants to write a grant for that, because he called me, was it, I think some month ago, that, oh, let's come and work on this proposal together, since you are into the CRISPR, he's into the mosquito study, so let's just collaborate and then write something on it so that we can, get something. So the idea is not just to use it the way we are using it in agriculture, or the way we are using it in the medicine stage. No, in this case now we want to introduce a kind of change in pattern of inheritance. A typical example now is I want to distort. Of course, we said the female mosquito causes the malaria, female anopheles, right? So the gene drive is going to help you to distort the sex in such a way that you might, in a controlled population now, emphasis on the word controlled population. So I'm not saying generally. In a controlled population, you want to have more of male mosquito than female, so that you reduce, the end goal is to reduce malaria burden, because more female mosquito meaning more malaria. So you have a controlled population whereby you have just more of male and maybe few female, so that it can reduce the burden of, because I mean, if you knock out every female, who is going to be giving birth to them? Of course, nobody. So that's why I said more of male than few of female, so that those female can just be used for reproduction sake and all of that. So it's not going to be, it's going to reduce the burden of malaria. So you're going to change the, of course, we should be all familiar with we should be familiar with Mendel pattern of inheritance, where we have your law of segregation, law of dominance, law of what's the third one now, independent assortment, if I'm correct. So we're trying to use the gene drive to take it away from that normal Mendelian inheritance to another pattern of inheritance, so that we don't just follow the normal inheritance pattern. We are now following, we have distort. The last study that was done on it, I think, in one of this African country, they use it to distort the sex of that particular controlled population. You get the point now. So that is how important the gene editing tool can work for you. So you can just use it to, and there is a what I say, a worldwide proposal now, that maybe they want to deploy it in Kenya at the moment. That they want to you do a gene drive so that they can reduce the number of females of mosquito. Don't let me just say female. They're going to use it to distort the particular female gene so that we have more of male, and it's going to help us to reduce the malaria burden. So, and lastly, we have bioremediation, which is using a genetically modified bacteria to degrade plastic. And my professor told me that we should write on this, but instead that We're still trying to do some literature review on how we can go about it. You can use genetically modified bacteria to degrade plastic. Of course, one of the problem we have in Nigeria, so to say, is the plastic pollution. So using though it takes longer time, engineer those microbes in such a way that whatever gene that is causing them to whatever gene that is causing them to degrade the plastic, we can upregulate the gene. Please, are we together at this? All right. So whatever gene that is responsible for them to be able to there. So you can imagine if the gene is being degrade more or less that you're trying to fast track them to do that. Gene. Now let's talk about the next one which I'm going to be talking about, which is the common troubleshooting in gene editing. And one of the most we are, which I think I've talked about that before. So what can cause your gene editing efficiency? Number one is poor guide RNA design, which is why I try to explain RNA design part very well to us. So if your RNA design, maybe your efficiency is very low, or you just picked randomly, you pick the one that is good. You pick the one that can cause your guide RNA not to be effective, not to guide the Cas9 to where it's going to cut. So it's going to lead to low editing efficiency. So the guide RNA, you have to redesign what you ought to pick. And then number two is your weak promoters. In your plasmid, if your promoters are not, your plasmid is for expression. So if all of those things are not well expressed, it's not going to help you. So you might probably consider a new plasmid. The third one is your inefficient delivery. The inefficient delivery means when you are transferring into the use, is it that you don't use zero resistance, they call it now, resistance or whatever. Is it that you don't use the buffer you ought to of the plasmid into the cell line? So you have to check the vector as well. So if your plasmid is not working, then you can choose another plasmid. Plasmid is just one of many vectors that is associated with gene editing experiment is off-target effect, which can causes the Cas9 to bind to unintended sites, and this mismatch that is giving you signaling like 2-3 times, then that means your Cas9 can go and cut in different places. So how to use high fidelity Cas9? Like I said, the Cas9 is just an enzyme. The same way you have FOC1, the same way you have Indo3, the same way you have SMN1, the same way you have, and those enzymes, especially if you have done restriction. An example now, Apple1 is a very good enzyme I use in the lab if I want to study a particular multi-drug resistant gene in the plasmodium. I use Apple1 to digest the PCR products. So the Apple1 can be normal Apple1, and I can decide to use another one, the Apple than the normal Apple1. So in this case too, now you can have ordinary Cas9, that will be HF Cas9. The HF Cas9 is much more powerful than if your ordinary Cas9 is giving you a problem, then you can switch to your high fidelity Cas9. Then the rigorous bioinformatics validation, which I think I told us that the sources of your experiment depends on how good you are on the computational stage. If your computational experiment is failed, then you are likely, or if you don't really give consent about your computational aspects, then you are likely to have a failed experiment. The next one is your delivery challenges. Are we all together now? Are we together? Okay. All right. So we have the delivery challenges, which is the which vector you want to use. Like I made mention, or let me say which delivery method you want to use. Like I made mention, we have electroporation, which is using electroporator to carry deliver those plasmid inside the cell using electric field. We have nanoparticles, and nanoparticles is what my professor current student are currently using. We have viral vector. We have viral vector, which is AAV, adeno something virus, and we have a lentivirus. So these are different methods. The lentivirus is mostly used if you want to do CRISPR high. There's another CRISPR, like I said, CRISPR-Cas9 is very versatile. So there's another aspect of CRISPR-Cas9 that is called CRISPR high, CRISPR interference, which you're trying to do a CRISPR repression. If you notice when we were trying to design a guide RNA yesterday, when I was trying to pick an option, you can see CRISPR-Cas9, repression, knocking, knockout, all of those things. So in the repression, that is the CRISPR-I, CRISPR interference. So in most of those cases, the plasmid you use, would I say the vector you use for them is called a dCas9 KRAB vector. dCas9 meaning deactivated Cas9 KRAB vector. Remember, in that case, you're not cutting per se. You are just trying to repress the expression of the gene. So in that case, you can use your lentiviral vector. You can use your lentiviral to deliver them into the cell line. So you balance between efficiency and toxicity, because if your plasmid is toxic, it's going to lead to a problem inside the cell. Like I said, you're trying to solve a problem and you're giving room for another problem. So the next one, which is the last one, okay, not the last one, cellular toxicity and DNA damage. The excess Cas9 activity will cause apoptosis. Of course, we know what apoptosis means, which is programmed cell death. If the Cas9 is too toxic, just to be cutting. I'd use an example of the blind man. If a blind man is just swinging the sword anyhow, maybe he's angry, he's being toxic. If care is not taken, he's going to kill everybody. He's going to anything that comes across his way, take them down. So if the Cas9 is very, very toxic, excessive of the Cas9 can lead to apoptosis, which will cause the cell to die. So you have to use inducible system that can induce the Cas9, repress the quote unquote the too much expression of the Cas9 activity, and reduce the dosage. So these are the common troubleshooting that I have experienced, and as well some of the things people in CRISPR-Cas9, the field that they've experienced, and how to go about it. Remember, I don't just give you the trouble. I don't just give you the problem. I give us how you can, the possible solution to heat, which I don't want to repeat everything again, but I believe we understand from the low eliding and efficiency, the causes, the solution, aftercare, the causes, the solution, the river challenges, and all of that. So that is going to be that for that. And then another part we now need to take consideration is the future trend in gene editing. Like I said, it's called building the researchers. It's not just build. There's difference between. I mean, I'm not a grammarian, but I want to believe at least I know English more. There's difference between build the researchers and building the researchers. So when we are saying building, is a continuous work. If we want to build the researcher, we'll just limit ourselves to the what I say, the understanding that we have here at the moment. So it means we won't be able to. We just train you just for now, but it is called building. It is in progress, continuous work. So Why that is important, if it is just about build, we wouldn't need to be talking about future trend. But because we are building, that is why we bring in the future trend, so that as you're learning, you keep updating yourself, you keep upgrading yourself as well. So the future trend in gene editing, one of them is base editing. The base editing is just like you're trying to use a single nucleotide conversion without double strand break. So in this case now, of course, CRISPR-Cas9, you still have to, there should still be a double strand break. But in base editing, you're not going to experience any double strand break, which makes your work very easier. Especially if you want to study SNP, you just convert that particular nucleotide without you subjecting them to maybe a double strand break, or what's it called now, or HDR, or non-homologous end joining. So it's just going to convert maybe your G to C, or convert your T to A, depending on what you're trying to do. Okay. So the next one is your prime editing. The prime editing is very versatile editing with reduced risk of unintended change. So all of these things, although they have been used, but it's not as used as the CRISPR-Cas9 technology. So it's still ongoing work around them, so that we can really see how far we can go. So the prime editing is one of the upcoming gene editing tools uh, that is being associated with a reduction of unintended changes, especially the toxicity of your Cas9. So it's going to help you to reduce all of those burden. Then the next one is epigenome editing. When we're talking about epigenome editing, we have to talk about epigenetics. If I may ask, does anybody know what epigenetic is?
Okay, sir. Can you hear me, sir?
Loud and clear.
Okay, so I think epigenetics is referring to environmental factors surrounding the genome of the organisms that can affect the gene and the gene expression.
All right, thank you so much, Olajumake. I think Olajumake is my next favorite after Bimbola now. I don't know why guys here are not talking. It is well with you people. Okay, so thank you, Olajumake. The epigenetic is just like you rightly said, the factors that are associated with environmental, circumstances that affect the expression of the gene. So in this case now, unlike your point mutation, unlike your frame shift mutation, that is change in the sequence. In epigenetics, there is no change, especially when you're talking about epigenetic mutation, epigenetic modification. There is no change in the sequence, but there is something we call histone modification or methylation. Histone modification or methylation. So these are the things that attach themselves to the gene without causing a change into the DNA sequence, but there is a mutation. So in such a case now, you don't want to use CRISPR-Cas9 to be, because you can only use CRISPR-Cas9 maybe there is a change of G to C, or there is a change of A to T. No. But in this case, you are not doing G to C, A to C, because the mutation that is going on is not even in the DNA sequence. It is due to histone modification, it is due to methylation. So epigenome editing is now coming to correct that, is to, would I say, to address that part. So you can be able to control the gene expression without you altering the DNA sequence. Like I said, epigenetics has nothing to do with your DNA sequence. So you are not altering, you are not editing the sequence. You are only controlling the expression by probably removing the methylation, or reducing the histone modification that is going on. So the next one is in vivo therapy, which we are delivering editing tools directly into patients for treatment, which I've told us a live example of somebody that one is being used for, Jimmy. It was delivered in vivo, and the guy is fine after 5 years. And I want to be sure that you guys must have heard about those two girls from China, which they used a gene editing to, you know, stop them from having HIV. And the guy was jailed, but though he has been released now. So these are the kind of in vivo therapy, not just using it to edit cancer cell line alone, not just using it to edit parasitic cell line immune. All of those ones are just study. In this case now we want to turn it to therapy so that we can put them inside, like and anybody having cancer now, just design, collect the sample sequence, get where this mutation, design your guide RNA, design the experiment, this one, that one, then transfer the therapy in transfer the tools into the patient, and the patient should be fine. The same way your cell line is fine, the patient should be fine. So it gives room for therapy. Anybody with sickle cell, people won't have to be worried. And I think by, was it 2023 or 2024 now, the UK being the first country to approve FDA CRISPR-Cas9 as a therapy for sickle cell. So you can see that means the in vivo therapy is even already ongoing. And the next one is a multiplex editing, which is, of course, the one multiple means you are trying to do something simultaneously, many things simultaneously. So you are editing multiple genes simultaneously to address complex traits, especially if you have a polygenic trait, which I want to believe our molecular biology mentor should have probably talked to us about all of those normal terminology. So polygenic traits, if you have that particular disorder in the polygenic traits, instead of you targeting them one by one, you can do multiplex editing, which is still upcoming as well. And the last one is your AI and machine learning, which makes it easier so that you can predict whatever you want to do online first. You predict the off targets, you design, optimize guide RNA, you do everything. And one of the optimizing of guide RNA, which I told us is the codon optimization. We couldn't do that today. Like I said, it's part of the design of the construct. That is going to take us a whole lot of time. So, like I said, this is just a foundation to this. It's not to turn you to an expert within just 30 days. No, just if your interest lies in this, then we can take it off from there. So you don't need to worry yourself too much about, oh, we didn't do this, we did not do that. No, just get the basic one we are sharing, then you decide for yourself. Now, what are the ethical concerns in gene editing? Sorry. What are the ethical concerns in gene editing? Of course, like the first day that I asked one of us that, okay, what is gene editing? The first thing that came to the guy mind is genetic modified organism, and about the controversy. So that means that there's an ethical concern about it. Now, one of them is human germline editing, which raises the concern about designer babies on unpredictable long-term effects. That, okay, what can they now want to turn, they might probably be using to now make it a baby, artificial baby, or maybe if they use it to treat the patient now, what about the long-term effects? And I told us the guy that was treated 5 years ago now, the guy is living fine. He turned 40 last was it last week or yesterday now? He turned 40. So, which I understand that the concern is valid. However, that is a room for growth. Like I said, gene editing is not something that is as common as you have your PCR, you have all of that field. No, it's still upcoming and it's still gaining recognition one by one. So it is an upcoming tool, and we have equity and accessibility, risk of widening health and wealth inequalities. Of course, a typical example of this now is that when the UK approved the CRISPR-Cas9 to be used for sickle cell therapy, the question is, will it be available for everybody? Which the answer is no. So that's why we're trying to talk about equity. It's going to be only for rich class people. Take, for example, you have a poor people that is living with a sickle cell, and we have a rich people that is living with a sickle cell. Of course, they are only going to attend to the rich people because of their wealth. So that means the sickle cell man, the common man, has no hope. So he has to die. Even though the dying is dying now, it's not because the technology is not available, but because of the accessibility. It's not there. So that's another concern. So if only it can now be available for both the wealthy and the average person, then we can now say, oh, this thing is actually good, even though it is good. But the barrier about the wealth and the lack is making it is raising a concern, which is a public health concern as well. So the question now, who benefits from it? Is it the rich? Is it the poor? Is it so that is a concern that is valid. The next one is environmental risk. I made mention of gene drive. The other time that in gene drive, you are trying to change the pattern of the inheritance, maybe distort the female Anopheles mosquito gene. And there is a concern about it as well that if you say you want to distort the gene, that means you are indirectly saying you want to kill all the mosquito, because if there is no female who will be giving birth to the new mosquitoes, and some on the school of thought that mosquito is necessary for ecosystem, even though it's giving us malaria, some said it's still at least beneficial. So if you now distort them now, we don't have what will benefit, especially we in Africa, that their mentality that is still benefiting to us. So there is a concern about it. Some of the school of thought, oh, we can distort this because of the malaria burden. As a matter of fact, Nigeria is the highest country in the world with high global malaria rates, higher mortality rates rather. We are the first country in the world that people die of malaria every year. So some are in the school of thought that, oh, let's do this gene drive to reduce malaria. Some are in the school of thought that, oh, it's important for our ecosystem. So it's all about back and forth. And potential for biodiversity loss, which I think I've already explained. The consent and governance, it needs a strong regulation, oversight, and global consensus. Take, for example, when I facilitated in the workshop we did last time, some of the participants say, oh, now they can now go and be using CRISPR. I was like, no, if they catch you, they will arrest you, they will jail you. Not be me teach you. So you can't say you just want to do something and the next thing you just know you should be ethically grounded. That's why I said I have to emphasize this part, not just teaching you what CRISPR is, I'll be teaching you what gene editing is. You have to be ethically grounded. So there is need for strong regulation, and you don't just do gene editing anyhow. You don't just carry out the experiment anyhow. You have to be ethical concern, ethical, what do they call it now? Ethical approval has to be very, very strict, because we're talking about something that is still going on, or what I say, upcoming, and it still needs a critical attention. So you don't just want to do it and that is why the man in China was jailed and his license was sealed. So you have to be very, very conscious. So there is a need for strong regulation over it. And then the difference in cultural and legal framework add complexity, in the part of the culture, I would even say belief. Some will tell you gene editing is bad, that you are trying to change God's nature, you are trying to do this, or you are trying to do that one. There are some people of the school of thought like that, which is their own cultural belief that you don't alter what God has done. And these people forgot that the surgery you are doing, how is it different from gene editing? Of course, it's not different. So if you are in the school of thought that, oh, gene editing is bad because you are trying to change the nucleotide, you are trying to change the DNA the God has done, then why do you need to go for surgery as well too? Because at the end of the day, they're both a therapy. One is the targeted therapy, one is the surgical therapy. So there's still be both therapy at the end of the day. So why do you now try to demonize one and try to now paint one as an hero? So those are still one of the concerns as well, which is still ongoing debate, here and there, which I believe with time, all it just needs is okay, I think I have it here now, that the possible solution which cannot be done is the what I said, the most important one is public education and awareness. Scientific progress must be matched with ethical consideration. It ensures transparent and regulation clinical trials, promote international cooperation and safety guidelines, and public education to reduce misinformation and build trust, which is the emphasis. We have to make sure that all the misconception people have when you hear gene editing now, they will automatically think it's something evil, something you want to do. They forgot that the surgery is almost the same, is even the same. I mean, people do surgery and still die. So what's not the point? So we have to correct that misconception. We have to correct that misinformation of ideas, so that when they hear gene editing, they won't just think of something demon. They won't think of something evil, so that we can build trust. I mean, I want to believe when surgery starts, it doesn't just start on the platter of gold. There are some concern about it before it now became one of the most, therapy approach. So it gained trust along the line. The conception was changed. The same thing going on now, which I believe with time to all of those. And what is just required is public education. Teach normal people what they need to know about gene editing, so that they don't just carry this perspective or it is an evil stuff. So once we can able to do that public awareness, public education. It's going to reduce the misinformation. I'm using the word reduce, not cancel, because of course you can't change everybody's mind. But at least if you have a good percentage of people, till today, some people are still of the opinion of surgery is bad. And in respect to, I mean, in no disrespect to anybody's church, there are some particular church that will tell you even blood transfusion there is against their belief. So that's why I don't want to use the word cancel. You can only reduce the belief, you can't cancel it. So so that at the end of the day we can have innovation that is safe, equitable, which is okay for both the poor and the rich, and the socially responsible environment. So this would be the end of my class. My question is, do you want to become a genome editor? You have to now make the decision. And it's fine if you don't want to be. Gene editing is not just the only way you can become a researcher. It's just one of the way. So it's not something that, oh, I must become. No. So if you want to be, then you just have to make a decision for yourself and position yourself for it. Thank you, everyone.